All right, we are right at 9 o'clock GMT, so I think we are going to go ahead and get started. So welcome everybody, and thanks so much for joining us this morning. My name is Willie Goldsmith, and I'm a Senior Associate with the LenFest Ocean Program in Washington, D.C. And for those of you that don't know LenFest, we are a grant-making program that funds research projects and expert working groups to address priority science needs that are facing ocean and coastal decision makers worldwide. And if you'd like to learn more about our program, you can visit us at lenfestocean.org, where you can also sign up for our monthly newsletter, and you can also follow along on Twitter. We are at lenfestocean is our address. And for today, we're really excited to have with us Dr. Mark Bravington from Australia's Commonwealth Scientific and Industrial Research Organization. Many thanks to Mark for staying up a bit late tonight to accommodate our Eastern Atlantic audience. Thanks for that, Mark. And he'll be sharing preliminary results of his work looking into the potential for an approach called close-kin mark-recapture to improve estimates of Shortfin Mako shark abundance and mortality across the Atlantic Ocean in both the North and South. Mark and his colleagues in Australia have successfully applied this approach to other species, including some shark species. So we at LenFest, we're really excited to support Mark's work to look into trying this technique with Shortfin Makos. And I'd also like to note that the Shark Conservation Fund is also providing additional funding for this project. So before I turn it over to Mark, I just want to give a couple quick logistical items. First off, as you may have noticed, we have all attendees muted just because, we want to prevent any feedback or anything of that sort. So just be aware of that. After Mark's talk, we'll have the last 20 or 25 minutes or so for questions. And if you do have a question, you can use the Q&A panel in WebEx to type and submit those questions at any point. And I will keep track of the queue and read those questions aloud at the end for Mark to answer. And we may or may not get to all questions, but hopefully we do. And if not, of course, feel free to follow up with me. My email address is on the screen here, and Mark's email address will be on the closing slide as well. And we definitely encourage you to reach out as we kind of think about, next steps for this exciting project. I do want to mention that we are recording this webinar, and hopefully in the next couple of days, we'll be able to make that recording with the PowerPoint available online, and I'll distribute that via email. I do have a running distribution list of everybody who's registered, and so hopefully, you are on our distribution list, but if you want to make sure, you can always shoot me an email, and I can confirm that you are on our radar. So with that, thanks again so much for joining us. And Mark, I'm going to pass presenter privileges over to you.
Got it.
Bear with me for 1 second here. Okay.
All right. Can you just confirm that you can hear me okay at the moment?
We can hear you great, Mark. Thank you.
All right. Okay. Let's get that going. And can you confirm that you can see that okay? You should be seeing a shark right now.
Yes, sir. We see it. Thank you.
It's a Mako. All good. All right. Well, thanks very much. This is a new adventure for me doing this. It's not actually that late at night down here. It's only 8 p.m. I can usually make it for another hour or so. I don't know if you can see my mouse pointer, but I'm actually doing this from my living room in Tasmania, which is this little dot at the bottom of Australia down there. And you'll also see that there's no word genetics in the title of this talk, but the stripy stuff on either side of the shark logo here is DNA, and that's critical to what I'll be talking about in tonight's session, or this morning's session, wherever you are. So there's really 4 parts to the talk. I'll start off with some background around Mako sharks, particularly in the Atlantic. The short summary of that is that they appear to be in a lot of trouble due to fishing, and it's a bit hard to tell what's going on. Close-kin mark recapture, as Willie said, is a technique that may prove very useful, and I'll go through the principles of that quite briefly and one or two examples, and then turn to the actual results of some design and planning work that I've done for Makos in the Atlantic, and come on to a summary. Could this actually work? Well, I won't give you a spoiler. I won't give you a spoiler alert at this point, but you know, you should be able to get the general drift. I will also say I've not done this without a live audience before, and it's quite a strange sensation. Even the cats have been locked out of the living room, so no feedback is an interesting way to do this. And I'll get the disclaimer out of the way right at the start. You'll work this out anyway, but I'm not a Mako shark expert. I'm not a shark expert. I'm also not an expert on the inner workings of ICAT. So I've done my best to get the broad basis of things right, but if you want to check the details, you need to go back to check them up with ICAT itself. What I do know a bit about is abundance estimation and fisheries assessment and stuff like that, and about close-kin mark recapture. So a little bit of background on Mako sharks. They live everywhere in warmish waters, but there are separate populations in different ocean basins, and in particular, the North and South Atlantic appear to be genetically separate populations, even though there's no clear boundary between them. Mako sharks shortfin Mako sharks, I should say, seem to stay out of the very warmest water masses in the middle. So there's genetic separation there, and they're managed as separate stocks in the Atlantic. There is another species, by the way, longfin Makos. I'm just going to keep referring to Makos throughout this talk, and the focus here is on the shortfin Mako species. There's probably a little connectivity between the Atlantic and the Indian Ocean, but sharks and Makos can sneak around the bottom of South Africa, but it's certainly not genetically panmictic there, so it looks again as though there's population structuring there. They're widely caught on pelagic longlines and have been for a long time. Unfortunately or fortunately, there's a high market value for their flesh. They're quite desirable eating, in contrast to a number of other shark species, which kind of means we have better data on them, but also, of course, people are very happy to catch them. And there's also, of course, the market for fins. So whether you call Makos a bycatch species or a target species is a bit of a question of taste. Certainly in most fisheries that do catch them, I don't think people mind very much that a Mako is taking up hook space because there's a good market for the flesh. A few Mako facts there. The salient points about them really, they're a late maturing species. Females don't mature till about age 20 and males around age 10. Males don't grow as big as females. Litter size 10 to 20, average is around 12, and bigger females have slightly bigger litters, but it's not a massive effect there. Breeding cycles are probably about every 3 years, so they're not a highly productive shark. They're not the least productive shark, but that late maturity does mean that they're not really cranking out the offspring particularly fast. In the Atlantic, they've been fished for a long time. There are separate catch series which ICAT has worked hard to produce in the North and South Atlantic. The North got going a bit sooner. That's the line at the top. And you'll see that recent catches have been in the range of 3,000 to 4,000 tons, closer to 3,000 recently. And that's about 87,000 individual sharks caught in 2015. Catches in the south have not been quite as high, but the catches are catching up, and the trend is still going up there. The important thing to note about the catch is that it's mostly immature sharks that get caught, ages 2 to 10. About 3% of the catch numerically is youngish adult males from ages 10 to 15, say, and there's hardly any adult females are caught. So ICAT has been looking at Makos for a while, and it's had a couple of goes at stock assessments in 2008 and 2012, but quite a few problems getting the data together. I think they're now fairly happy with it, with things like the catch series, perhaps because shark catches are often a problem to estimate because they're not well recorded, and so many animals are discarded. But people have generally not discarded Makos very much, precisely because you can sell them. So there's been more work on the assessment since then, and in 2017, ICAT came up with a defensible, full-on stock assessment, age-based, CPU-driven stock assessment. And some of the results out of it, there's an assumed, basically it's an assumed value of natural mortality of 0.08. These abundance estimates are worth bearing in mind. About 140,000 adult females, mostly quite old by now, and nearly twice that adult males because they mature younger. So the really alarming things about the assessment is how high the fishing mortality is compared to what a sustainable level might be. It's running about 4 times higher, 400% of the MSY, estimated MSY fishing rate. Spawner per recruit ratio, that's how much spawning an average, if an animal makes it to 2 years old, how much spawning is it going to get to do compared to what it would get to do if it was likely left alone for its life until it died naturally, is down at 21%, which is extremely low. It would be a very worrying figure for a teleost and for a shark, which are less productive. It's a real worry. So the stock appears to be overfished. It's less clear in terms of how depleted it already is, but the level of overfishing is extremely worrying. There are some projections from the assessment. Let's not spend too much time on them. They don't actually look too bad, but that's because the projections that you're seeing on that plot all involve a substantial reduction from current levels of fishing and catch. The highest TAC they considered was only a third of the current catches. So some of the things to be concerned about is that whatever you do, even if you stopped all fishing tomorrow, the stock's still going to decline until 2035, the adult spawning stock, that is. The catch would have to be reduced, or the lethal catch, to about one-sixth of its current level to give a 50% chance of rebuilding the stock by 2070. And current management practices are not going to do it. In the South Atlantic, well, catches are still going up, as we saw, but so are the catch rates, the CPUE, catch per unit effort. There's no way that can make any biological sense, but one, it's not because the abundance is going up, something else is going on. But the upshot is no useful assessment is possible. So people are worried about it, but it's, the trend is going the same way as the North Atlantic, but there's nothing quantitative you can turn into a sort of hard assessment there. Some recommendations. There is now an ICAP policy of if you catch a live Mako shark, you're supposed to release it. And if that's completely followed through, that might reduce the fishing mortality by about 50% with no other changes in practice. There are some proposals that even dead sharks should not be saleable. We'll see what ICAP does about that in a few days' time when they have their commission meeting. And there's been no concrete proposal no agreed management action in the South Atlantic yet. Makos have been listed under CITES Appendix 2. It's not clear to me anyway what that will do to catches. It does have some other implications that we'll come to later, but they're certainly on the radar both of ICAP and globally as a species that's in trouble. So catch per unit effort. A few polite comments. If I was giving this talk in Australia, I might be a little more robust with my language, but it's going internationally, so I'll be nice. I think it's a great accomplishment that ICAT has managed to get these shark data sources together to the point where it can produce a defensible stock assessment. It's a very difficult thing to do for pelagic sharks because the underlying data tends to be not well reported. But there are still issues with that assessment. There are things you have to assume, like the natural mortality, and it's still entirely reliant on CPUE, catch per unit effort. And as you can see in the South Atlantic, clearly that's not telling the full story. I've worked lots on stock assessment over the years, and when I first came into it about 3 decades ago, I thought catch per unit effort was marvelous. And the longer you stay in the business, the more you realize that a lot of things can go wrong with it, and they tend to go wrong over time. There are lots of things that affect the catch rate. It's not just the abundance of animals. In a species with a big spatial distribution, such as Mako sharks, then holes in spatial coverage become a problem. What was going on there? Fishing boats move around in response to economic incentives as much as anything else, and so do fish as well. So you don't really know that you're covering the whole the whole stock, or that the proportion of the stock that you're covering is consistent over time. And of course, changes in fishing and targeting practice. Mako sharks are not the primary goal of most of these fisheries, so the way that the fisheries operate can change in ways that's got absolutely nothing to do with Mako shark abundance. On the plus side, CPUE is free, and on the minus side, you certainly get what you pay for. And even when it does work, it's still just a relative index. You'd really like to be able to turn it into absolute abundance, and that's a hard thing to do with CPUE. That's what stock assessment's about, and it's not easy. Really, if there was anything better, you'd rather use it. I should be quick here, but a couple more things that are a little bit specific to Mako sharks. All the catches and all the CPUE data is about immature animals, so there's no direct ability to track what's going on in the adult stock. And of course, that's what you want to look after in fisheries. Make sure there's enough adults around to keep pumping out the babies. So by the time you do start to see big falls in CPUE resulting from low recruitment, the adult stock must already be well in trouble. Something else to think about is what's going to happen to CPUE in response to management measures in future. Catch may not mean what you think it meant, and effort may not mean what you think it meant. So how are you going to tell whether any conservation measures that get introduced are actually working? I won't say anything much about mark-recapture here as another method, but it's a bit, I think, logistically infeasible to consider something like that for Mako sharks. So what are you left with if it's not CPUE? So now we're on to principles of close kin, and I'll start off with this cartoon diagram because it's easy to explain. This isn't actually what we would do for Makos, for reasons that I'll make clear, but it does get the principles across. And you have to be aware this is a cartoon. Real biology does not work quite this way, and you need to make it a bit more sophisticated. The point about this cartoon is that you've got some small pink fish in the sea and some they're juveniles and some big blue adults. And each juvenile has exactly 2 parents. That's the lines coming out from the juveniles into the adult population. Now the adults themselves, they can have a variable number of juveniles. Some won't have any offspring, some will have lots. But each juvenile must have had exactly 2 parents. And if you think about sampling a juvenile, well, what that's doing is that's tagging its 2 parents in the adult population. If you go out and check whether any one of the adults has got that tag, the chance that the particular adult you sample is 2 over the total number of adults that you could have sampled from. So that's just one juvenile. What you do in practice is you go out and sample a number of juveniles and a number of adults. There's 6 and 4 in this case, and I've left the other animals visible, but of course you wouldn't be able to see them in a real situation. Once you've sampled them, you can look at their genotypes and find out which one of them are parent-offspring pairs. You can do that genetically in quite a reliable way, and in this case you find that 3 of the comparisons that you'd make bring up parent-offspring pairs. So in all, we've got 24 comparisons that we could make between 6 juveniles and 4 adults. And every time we do it, the chance that it's going to give you a parent-offspring pair is 2 over the number of adults. So the total number of parent-offspring pairs, POPs, that you expect to find is 24 times 2 over n. And we actually find 3. So the value of n that gives you the best fit to the observed data is 16 adults. And of course, because I fiddled the numbers, the true value of n in this case is exactly 16 if you count the blue fish. In practice, it won't come out at exactly 3 parent-offspring pairs each time. And that illustrates the point that you need to make sure you get a fairly big number of parent-offspring pairs in your final result, so that variability isn't too much of a problem. It's pretty good. The health warning is that you can't apply this literally. There's more aspects of biology and sampling that you need to take into account, but that there is a systematic way that you can do that, which I won't describe in detail today. Now that's all about parent-offspring pairs, but the problem with that is you need to be able to catch adults, and with Mako sharks you can't, at least not in useful quantities. So there's an extension of this that you can apply to looking at half siblings. A half sibling is a pair of animals that's a half brother or a half sister. It's two animals that have got either the same mother or the same father. And you want to do this by looking across cohorts, two different cohorts that are separated in time. The reason I don't talk about full siblings is you don't tend to get full siblings except when they're in the same cohort as each other, and that turns out not to be very useful. So in this case, we're going to sample only from the juveniles, and we're going to sample from two different cohorts. The samples are the sort of yellowy, orangey blobs you can see there. So if you can think about sampling one animal from the first cohort and one from the second, what's the chance that they're going to be a maternal half sibling? That is, do they share the same mother? Well, Lucy in the first cohort, we know she had one living mother when she was born. And the chance that that mother is the same one as Joe's, well, first of all, Lucy's mother has to survive the intervening years. And secondly, if she does make it, then she's got an equal chance as any of the other adults that are alive then. But the adult population will have changed in that period because some have died and some new ones might have recruited to it. So the point here is you can estimate two things this time. You can estimate adult mortality rate as well as absolute adult abundance. But to do it, you're going to need several cohorts to do comparisons, not just from A and B, but from A and B and C, and mix all these things together. And again, it's a cartoon. Reality is a little bit more complicated, but we can deal with those complications for something like Mako shark. So in practice, you don't just do individual cartoons. You'll sample for a number of years, and you'll sample, in particular, a whole bunch of different juvenile cohorts. So these grey slices that you see here are all meant to be, say, 1 year's sample, or different cohorts of juveniles, let's say. And we'd use exactly the same animals in parent offspring pairs over on the left, and the same juveniles, but not the adults, in half sibling comparisons on the right. Put everything together, and you do it all in a sort of proper statistical model way. You're going to compare every sample to every other sample. There's lots and lots of pairwise comparisons. And whenever you do a comparison, the chance that it's going to give you a kin pair depends not just on the population, but it depends on the particular characteristics of the animals that you're comparing. So in particular, if you compared an adult caught late in the study with a juvenile born in the first, well, yeah, potentially that could be a parent-offspring pair, because the adult might have been alive when the juvenile was born. But the other way round, if you compare an adult that's been caught here with a juvenile born several years later, well, if the sampling was lethal, then it's impossible for that to be a parent-offspring pair. So not all these probabilities are the same, and that's what actually lets you estimate things with close kin. And then ultimately, we're going to, after working out what the probabilities might be, we're going to use genetics to tell us the outcome for each pair and fiddle around with the demographic parameters, the abundance and so on, until we get the best fit to all those comparisons. The most important thing about it is that you can do all of this if you have to with dead samples. So it's very useful for fisheries work. But it's not compulsory to be dead. And it's perfectly feasible to do this from live biopsies as well. So we'll skip over this, but there's a standard way to put this all into something that looks like a conventional stock assessment and gives you the same kind of quantities. Very simple core assumptions. I'll say a tiny bit about the genetics later, but all you need to think about for now is that you can do this kin finding stuff reliably. And on to the first example. I'll keep this pretty brief. Southern bluefin tuna is now becoming quite well known as a close kin example. The adults spawn in that big yellow blob that you can see to the northwest of Australia, and juveniles, when their age is 2 to 4, let's say, they hang out in that big red blob south of Australia. And they're caught in both places. And in the intervening years as subadults, they're caught in longline fisheries in the blue area. They're quite a late maturing teleost, and the conventional data on them is very challenging. So in the early 2000s, there were huge problems trying to put a stock assessment together because the CPUE data was full of holes and changes in fishery practice that made it uninterpretable. What's more, it was revealed that there had been massive overcatching of this species going on for about 20 years. So, who knows what that actually meant for the catch data? That was certainly wrong, and the CPUE as well. So it was heavily depleted, but no one had any idea what the abundance was. So that's where we started doing close kin, and we genotyped a large number of adults and juveniles very laboriously with the techniques that were available at the time. And it gave us about 38 million pairwise comparisons that we could do. And in the course of that, we found 45 parent offspring pairs. Got a whole bunch of results about it. I think the most important to focus on here is simply that we were able to reconstruct an absolute time series of spawning stock biomass for the cohorts that we sampled, which was quite different to what the assessment had been telling us. There's no way that the numbers in the assessments could have been compatible with what we saw in these data. But that's not really a surprise, because no one had any idea what numbers should really be going into the assessment in the first place. It worked very well, and so we've continued the sampling, and we've now changed our genotyping technique to next-gen sequencing, one of the varieties of that. And our motivation, apart from cost and so on, is that now we can find half-sibling pairs as well. We've updated the model with half-sibling pairs, and we've basically got now fishery-independent estimates of adult stock. And the CCSBT, the commission that looks after southern bluefin tuna, is now building this directly into its TAC setting rules for the future. So in the interest of time, I'll skip over this, but southern bluefin tuna has gone from being a bit of a joke internationally in tuna management to now what I would say is clearly the best managed, best scientific and politically actually managed of all the large tunas. And in part that's due to having these really sound data from close kin. We've actually done a whole bunch of close kin projects, though. We just haven't published them yet. So I think there's about 6 or 7 species in all. Most of them have turned out to be sharks around Australia for sort of accidental reasons. There are a couple of studies that have been done outside Australia as well, but not by CSIRO and CSIRO. And there's a whole bunch of studies starting up worldwide now that hopefully will yield results in a few years' time. I will point out that a number of these were done with live biopsy sampling. That means in particular we wouldn't know the age of the animals, but we were able to do a reasonable job based on their length. And I'll just mention quite briefly another example of besides tuna, which is school shark, because it's a little bit similar to Atlantic Mako. Again, like Mako's, few adults are caught of school shark. They're quite late maturing. Heavily depleted in the 1990s, so they're now basically a bycatch species with very tight catch restrictions off southern Australia. And the problem from a scientific management point of view really is that the CPUE series is no longer informative because the fisheries had to change its practice to avoid these animals. So how can we tell whether it's actually recovering or not, which the industry claims that it is. So we've done a full age-based stock assessment with close kin. 3,000 samples, 65 half-sibling pairs. We have an absolute abundance estimate now. The trend estimate is not yet significant, but all we need to do to determine what's happening to this stock is to keep sampling, and that is going on, and we'll be able to update the assessment in future. So overall, there's 5 things you have to get right in these close kin projects. There's no magic in a sense to any of them, but if you don't get the details right, the whole thing will fall over. I'm undecided in my own mind as to which of these words best describes close kin. One important point is that they're pretty sizable projects. You have to get the details right, and there's a lot of specialist expertise. So to cover off on them, you really need to have a team. I can't do the details of the genotyping or the sampling, that's for sure. One slide only on genotyping, I'm afraid. So any geneticists tuned in are going to be a little bit disappointed. The salient point here is that you're going to end up doing an awful lot of comparisons in close kin, tens or hundreds of millions between all your samples. You're only going to expect to find maybe 100 true relatives in the course of that. So you need to keep false positive rates, well below the 1 in a million mark. And in the course of that, you are going to incur some false negatives, some relatives that were present, but you just weren't able to detect them because you had to be so strict. But it can all be done if you pick the right genotyping technique and methods of analysis. You don't need to settle on this at the outset, but everybody involved in the project does have to do it the same way, and it's best if all the genotyping is done centrally. We have a method that we're relying on now at CSIRO, and it's worked pretty well for us, but we're always working to improve it. So coming on to design for Makos at last, what do you need to ensure to make for a project to work in the North or the South Atlantic? Well, you need to have an adequate sample size, because if you don't have enough kin pairs at the end of this, you're not going to be able to make any kind of estimate. And you need to cover off on these other points that I've listed there. So really the question is what's going to be adequate and how do you determine it? The quality of the final analysis will be set by the number and the pattern of half siblings found, for instance, if there's a good spread in cohorts. And in order to control that, you have to change the sample size. That's the one thing you can control in all this. So you start with a demographic parameter estimates coming out of something like a stock assessment. To go through the design process, I needed to build an assessment model that would pretend to take close kin data in future, and then given a potential design, sample sizes and so on, you can use that to work out how precise the things that you're interested in would be by simulation. In fact, you can shortcut that, but that's a statistical detail that's really only of interest to people like me. And then you mess around with exploring different sampling setups until you've got a satisfactory result that's precise enough and not too expensive. So I built an assessment model for Makos, and just to underline the point, you can do this estimating natural mortality as well as absolute abundance, and also estimating something about the stock recruitment curve. I had to worry in this case about the details of the errors involved in estimating age from length, because we may not have length data for many of the age data rather for many of the samples. That needs a dead animal. And I didn't use any catch per unit effort at all in how accurately this would work in future. So a few things you can consider in a design: how many years, what's the total number size, do you go for big animals or small ones, and can you get age data for them? I know from other studies that we've been involved in that if your age data is just too bad, the whole thing does tend to fall over. And various different quantities you might be interested in. So here are some preliminary results, I should say, results from the North Atlantic. I'm going to put a lot of numbers up, and you really shouldn't try and read all these. A bunch of different sampling scenarios that one can look at. These are all, most of them set around a sample size of 5,000, but for varying periods of time. And one of them, I experimented with doubling the sample size to 10,000. They should all give, if the assessment is to be believed, a decent number of half-sibling pairs, 80, 90, 100. These are all good numbers to be building models with. And if you double the sample size, it's a standard property of close kin, you're going to get 4 times as many, not twice, but 4 times. What kind of CVs might you get on abundance? Well, they're pretty tight, actually, in the range of 15 to 20%. Obviously lower if you have a bigger sample size. And even a good ability to estimate essentially, you could call it the productivity of the stock, or the spawner per recruit ratio, I suppose, is more the, how the fishing is going at the moment relative to what the stock could stand. So pretty decent CVs. Most of the options, except for sample size, don't affect the precision all that much. Even to my surprise, how many animals you age. So it looks like you'd be able to get away with length data in this situation, which is important because it's a lot easier to measure length than to take a vertebra from a dead shark. Short studies are a bit worse for trend, but you can always extend them. And what if the assessment is wrong? Well, we'll come to that in a minute or two. Really, these results are just meant as a starting point for detailed conversations about whether the whole thing is feasible and how to go about it. In the South Atlantic, we've got a problem because there's no assessment. So where do you start? And the solution I adopted was to say, well, let's suppose that the current fishing is just on the margins of sustainability. So a criterion that might be sensible for Mako sharks is spawner per recruit ratio of 0.7 or maybe 0.6. If that's where we are now, we'd want to have a study that would give reasonably precise estimates. So I reversed engineered a stock assessment that would leave us at that situation. It might not be true, but you wouldn't want to design a study that's going to be unable to perform in that situation. A couple of things to say there. What about if the assumptions of this exercise are wrong? Well, this is a nice thing about close kin. If you've designed it and you've been pessimistic, in other words, if there's a lot more animals in the sea than you think, then you won't find many half siblings. You won't be able to build a good model, but it's clearly good news. Things are better than you thought. And if you are optimistic, then you'll have lots and lots of kin pairs, and you'll be able to build a great model and know precisely how bad things are. Lots of numbers to not read for the South Atlantic. I think the bottom line is here, sample sizes probably need to be a bit bigger than the North Atlantic, but it may well be worth just trying and seeing what you get. Now, 10,000 is a fairly big sample size. You can take as long as you like to collect it, but in the South Atlantic, it might even be worth, if you only think you can get to 3,000 in a few years, it might still be worth doing because the stock might be more heavily exploited than the assumptions behind this design. Spatial coverage, I'm not going to have much time to talk about this. You have to be a little bit careful with spatial issues in close kin because they can cause problems. But Mako sharks do move a lot. They've been tagged as going right across the Atlantic, but not quite as much as blue sharks do. So they tend to stay on one side or the other for a few years. I think the bottom line is here that if you did all your sampling in one place, you'd be in for a problem because you wouldn't be able to distinguish between animals leaving and animals dying. But as long as you sample basically getting a reasonable number in the east and the west of the Atlantic, then any important spatial patterns, you'll see them in the data and can adjust the model accordingly. It means that no one country could do this. The US couldn't do it alone, for example, but it probably only takes 2 countries to get reasonable sampling together. You don't have to sample everywhere. Or evenly for a species that moves about a lot. Well, we're coming very close to the end of time here, so I better skip over this. This is why close kin's good. We can come back to that if anyone has time in the questions. To reiterate, it's very useful to have total catch data, but even if that goes haywire in future, it's not essential. You can still produce absolute abundance estimates and see how the stock is going. You don't require catch per unit effort data, and close kin is tamper-proof. There's no way you can basically manipulate it. So I presented the stuff at the Shark Working Group at ICAT this year in September. They had been talking about the possibility of close kin in the previous meeting, and they were quite enthusiastic. The results will be very useful for Mako management, both in the short term and also for monitoring any response to possible future management measures that ICAT might bring in. Looks doable on paper. Catches are not that high relative to what actually gets, sample sizes are not that high relative to catches. But lots of logistic issues still to sort out, including one administrative issue, essentially. There are some problems with being able to get samples now as a consequence of the CITES listing for Mako sharks. So I'll simply put up a paragraph that was written by the Shark Working Group in, I think, 2017. This issue probably attracted more comment than anything else I talked about in the meeting. Everything else seemed kind of doable, I think. And next steps in all this, as I say, the Shark Working Group was enthusiastic, so it's on their agenda for the intersessional in mid-2020. And there's also a tuna RFMO bycatch working group meeting in December 2019, which I'll go to with a couple of colleagues. And it's a good opportunity to get this, I think, onto the radar globally, particularly for pelagic sharks. This obviously isn't just an issue for Makos in the Atlantic. But in the Atlantic, we'll be looking to identify partners, and I think in that intersessional meeting and the run up to it, to develop a full program design in 2020, and maybe we can start sampling. There's worldwide interest, so one of our tasks at CSIRO is to try and develop tools for more general use of this technique. This has a last slide flavor to it. And indeed, I'll just finish with a few acknowledgements. The data for this provided by Enrique Cortez, the chair of the ICAT group and colleague at NOAA. To my colleagues at CSIRO that I've worked on with all these modeling and genetic exercises over the years, and to funding for this, which, as Willie said, came through Lenfest Ocean Programme and Shark Conservation Fund and CSIRO itself. So thanks very much.
Great. Thank you so much, Mark, for that presentation. If you wouldn't mind passing presenter privileges back over to me, and that way we can put your contact info up on the closing slide in case folks you know want to be in touch with you at any point about some of this work. But thank you for making the daunting tasks or the daunting issues of both population genetics and stock assessment science rather accessible in that coding presentation. So thanks for that. So folks, if you do have any questions, just a reminder that you can type those in via the Q&A panel in the WebEx tab there. So I'll just take a look at those, and I will share my screen. Great. All right. While we're waiting for some questions to come in, I think maybe I have one, Mark, to get things started, which is, you know, you mentioned the applicability of close kin for southern bluefin, and you know, I'm wondering, we're dealing here in the Atlantic with a with a data poor stock as a bycatch species, and I'm wondering, what did the findings of the close kin analysis for southern bluefin show, and how do they compare with previous stock assessments or other abundance estimates for the species?
Yeah, I skated over that perhaps a little bit quickly. I think it's now become so embedded into how the assessment of southern bluefin tuna is done that it's a little bit hard to look back at exactly how things were in about 2010 in the dark days. But one thing it did was eliminate a whole bunch of historical scenarios about what the catches might actually have been and what the productivity of the stock might have been, because at that point really SBT was data poor because of the problems with the catch series. No one had really any idea what the true catches might have been, and you know, if the catches are being misreported, which they were, then who's to say what the CPUE data would be? So the first thing I think it did was to knock out a whole bunch of implausible scenarios that were on the table and had looked plausible until close kin came along. It also tightened up a lot information about which fish are doing the breeding. It turns out to be that the big old females are disproportionately contributing offspring. So that has some implications for how long the stock's going to take to rebuild. And it so happened that the adults we were picking up with close kin had in fact been tagged with conventional tagging as juveniles at the start of their life. So rather fortuitously, it tightened up the assessment massively. And I guess that's why it's now, you know, its first job was to tighten up the assessment and eliminate some impossible scenarios. And the job it's doing now is to act as a monitoring tool in future to make sure the spawning stock does continue to rebuild. Because it was in a very bad place, SBT, and it's now rebuilding steadily, but it's still not a big stock. Yeah.
Great, awesome. Thanks for that. Thanks for that context there, Mark. We do have a question that just came in. Great talk. Can you provide a rough cost for doing the genetic sampling, not including labor, but just kind of the rough cost of genetic sampling per shark?
Yeah,
I can. About 5,000 sharks and how much that would cost.
Yeah, exactly. I actually had a whole bunch of notes written down that things I was going to mention during this talk, and I completely forgot to look at them as I was going through it. Our current costs are about inside $25 US per sample for processing and genotyping. So you know, it depends a little bit on how much things like handling and postage start to become an issue. But you'd be comfortable within US 30. So actually pretty cheap from that point of view. 5,000 samples, $30 a sample. I think you can work that one out. I'm always a little bit hesitant to put explicit costs in there, because in the end, the people time and so on will be considerably more than that, I expect.
Got it. Perfect. Thanks, Mark. Another question. Are there standard methods to validate close-kin mark recapture other than comparing them to more traditional assessments, kind of like what we were talking about with southern bluefin?
Well, I always have a smile when I see this, because, you know, what on earth in fish stock assessment would you take as a gold standard? I think what you have to do basically is be really careful about checking the assumptions. I mean, the core assumption is, you know, everybody had a mother and a father, which is pretty difficult to argue with. There are some things that can go wrong if you're a bit sloppy about how you translate the biology or the sampling into the equations. But really, you kind of have to think about it from first principles, and then it does make a lot of sense. It's not actually a magical process when you start to think about what the code might be. All of that said, one of the two pieces of work that I mentioned that is done by somebody other than CSIRO is some work on brook trout in Canada in a bunch of streams earlier this year by Daniel Rosanti and colleagues. And he had individual mark recapture data that he'd done genetically, and realized that he would be able to use this to sort of compare with close kin estimates. And yep, the two do seem to come out pretty comparably. I think given the deficiencies actually in the individual mark recapture and some quirks of the way the close kin analysis were done, I think they come out just fine matching up with each other. But it's pretty difficult to think what a gold standard would be in this situation. Yep, over.
Great. Thanks, Mark. Another question more from the population genetic side of things. How many SNP markers are used for parent offspring or half sibling assignment?
Oh, sorry about that. Yep, I totally forgot to say, and I meant to. Parent offspring is not very difficult these days. So the limiting factor is really how many are you going to need to do half siblings. If you've got enough for that, you'll have no problem with parent offspring pairs. So we would aim for about 2,000. We choose our SNPs fairly carefully. I think because a number of them, fail to pass various QC checks, we're using, I think, between 1,500 and 1,600 for SBT at the moment. Wouldn't really want to go too much below that. We have scraped a bit lower than that with some of our less abundant sharks, where you don't need to be quite as stringent. Yeah, 1,500 to 1,600 will do it.
Great, thanks, Mark. Give folks a few more minutes for some more questions to trickle in. In the meantime, Mark, I got one more for you. You were chatting. You're talking about cohorts, and you're talking about. It sounded like the idea of cohorts. Is that kind of in your traditional fisheries term, like annual cohorts? Is that how you envision the sampling taking place? So kind of regular, like you mentioned, Eastern and Western Atlantic for spatial distribution, right. But would you view like annual sampling from each of those locations over time? Yeah.
So when it comes to the equations, if you like, everything ends up being backdated to when a juvenile was born. So all the calculations sort of have to be backdated, because that's the point at which its mother and father were alive. So the analysis ends up being done at a cohort level, and the less precise your information you have about age, the more difficult it becomes to do that. So that's why knowing something about age is quite important. But, and I apologize for not making this clear, sampling, you have a lot of flexibility. In the design, I'm just assuming that the sampling would continue for 3 to 5 years, because it's just sort of logistically unrealistic to get it all done in a year. There's too much stuff to sort out with putting things in tubes and just getting used to the procedures. But you can take several years to do it, and you just sample. You can choose to concentrate your samples on, say, bigger sharks or smaller sharks, and that has some impact on how precise your results will be. but only within a limited range. There's no particular, you just take what you get in a given year, make some judgment, you measure its length, and that gives you some information on what cohort that animal might have come from. And then when you take it through to the analysis, that's the information you use. But you have there's a lot of flexibility about how you do the sampling. So I imagine that the easy way to do it would be to get samples going from a couple of, 2 cooperating countries, run them for a couple of years, and other people may well join the party. The one thing I did mean to say is that, the not so small print on this is that you do have to have a decent sample size. It would be a mistake to go into something like this thinking, well, we're only going to be able to get to 1,000 samples in all. Let's just try it and see what you get. You'd get nothing. You'd end up with like 2 or 3 kin pairs or something, which is not useful for building a model. So you have a lot of flexibility about how you get to that 5,000. And of course, you can have a look at the results before you get there, and they might tell you something different. But you really need to plan ultimately to get to that kind of sample size. Over.
Great. Yes, that's helpful. Thank you. Thank you.
That's what I should have said in the first place, Willie. I apologize for leaving all of these things out, but
Not at all, not at all. We've had a couple other questions come in. One is kind of thinking, a bit broader scope, which is how much more difficult would it be to use this technique in a developing country where a stock assessment has never been done, nor any tagging work on movement patterns? So the example here would be scalloped hammerheads in Indonesia.
Right. Yeah, that's a good question. So I think, what would I say there? So number one, half of our Indonesian samples, half of our southern bluefin tuna samples, well, all the adults are connected in Indonesia. There is quite a good sampling program there that we've set up, CSIRO set up over 20 years really with Indonesian fisheries institutes, and a fair amount of training there. I think you can separate various of those 5 stages. When it comes to the modeling and the design calculations, really that's always going to be at least for the foreseeable future, that's going to be a specialized thing that you get somebody in who can write this sort of code and they just do it. It'll get easier, but that's really probably not going to be push button for a very long time, but that doesn't really matter. If you can make sure that just the field sampling procedures are reasonably well followed, and all that means is that you've got to take your tissue samples in a way that avoids contamination and that avoids DNA degradation. So the new methods are quite sensitive to, new genotyping methods are quite sensitive to having good quality tissue, but there's standard ways to do that. And then the other thing you need to be able to do is basically get a good estimate, a good measurement of how long the thing is. And honestly, if you can do those 2 things, make sure the samples don't go off and get a good record of how long they are, then how big the animal was, then the genotyping can all be done by a third party. I mean, we ship ours off to a company. We do some lab prep. We do our own lab prep. But the company would actually do that for us if we paid them. And the analysis can be shipped out. So in some ways, the sampling requirements are not that bad for, not that daunting, I think, for a developing country. It needs to be a partnership, right? No one person and no one small team can really do the whole of one of these projects. Over.
Great, thank you, Mark. And one other question that I know we've chatted about a bit internally, but did you submit a paper on these findings to ICAT or perhaps to the joint TRFMO meeting that can be shared or perhaps shared at some point in the future?
Indeed. I submitted a very draft version to ICAT, and I'm just in the process of cleaning that, but I'm not sure that it's not really meant to be fully available from the ICAT website yet. However, I'm just cleaning it up at the moment, and I will make that public before the Tuna RFMO meeting, which is in December.
Great. Thanks, Mark. Thanks for that. Fantastic. I guess, yeah, we'll give folks, we got about 5 minutes left. So if anybody else has any questions they'd like to ask Mark, feel free to type those in. I think I have one more for you, Mark, which has to do with, which has to do with the timing of your piece of this. So I think, you know, you mentioned a minimum of kind of a 3-year time series. You know, as you think about moving forward, like, I guess the question is, from when you have all the genetics data in hand to when you can kind of provide an estimate, how long does that process typically take place? It sounds like much of the modeling machinery is kind of done up front. Is that right?
Yes, I think so. I mean, we've got a bit better at that, and in doing all of these projects and doing, for instance, the design work for this one, you have to do quite a lot of the model building in advance. So, in principle, you could do all the sampling for this, I should say, within a well, you possibly could do all the sampling for this within a single year, because any 1 year's sampling is going to collect a whole bunch of animals of different ages and therefore born at different times. But in practice, I just don't think, there's always some field stuff that needs to be ironed out. And that's why I think 2 or 3 years for sampling is a bit more realistic. But then you could turn it around within 12 months of that coming in, yeah.
Great.
And I think,
yeah,
go on.
No, I was just curious in terms of, you know, in terms of, you know, as management discussions are happening and thinking about the potential for that. So that's helpful context.
Yeah. I think one of the things I wanted to try and emphasize, I had perhaps a little bit more time at the ICAP meeting. Well, not much actually, but there is flexibility. So I didn't want to scare people by saying, well, you've got to have 5,000 samples by next year. If it's going to take 5 years to get the samples, the one thing about Makos that's in their favor is the adults don't get caught. So they're going slowly down and down and down as recruits fail to come through, but they're not going to go belly up tomorrow. So it is possible to take a bit of time over the studies if it's necessary to do that, to get the samples in. But there is a lot of interest worldwide in pelagic sharks at the moment, I'm happy to say, after they've been neglected really for such a long time. So I think it's quite possible that other initiatives in other oceans will get going. So there'll end up being some overlap in what we're doing. Some of I think for all of the species that we looked at, the field collection, possibly not for SPT, but that was the first one. For all the others, the field collection took longer than the analysis because we'd got so many of the wrinkles straightened out before it was time to write the final button and write the final model and push the final button. 3 years plus 12 months would be completely fine. We could probably squeeze it in less if that was necessary to do.
Great. Thanks, Mark.
Sorry.
Thanks. And no, thank you for that information. And just kind of to the point of you know, greater global interest, there was just a comment written. You may know that the WCPSC is just starting to look into this, and there was a suggestion from Australia that CSIRO will be invited to speak at the scientific committee meeting late next year, and that hopefully the joint TRFMO meeting will speed up buy-in in that regard. So certainly a lot.
Yep, I was aware of that, and yep, I certainly hope that gets some legs too. Yeah.
Great. All right. Well, I don't see any other questions coming in. We're about 2 minutes shy of the hour, so I think we are good to wrap things up. Mark, I want to thank you again so much for providing some context and background, as well as some insight into the methodology of this work. And again, if anybody would like to follow up with Mark, his email address is here on this closing screen. And of course, if you'd like to be in touch with us here at LenFest as well, you can email me at wgoldsmith@lenfestocean.org. Again, thank you all so much for tuning in, and I will be sending out a recording of the webinar in the coming days as well. So thanks again, and have a great day. Thanks.
All right. Yeah, and thanks from me to everybody for turning out and listening to this. Yeah, so sorry for the things I forgot to say and for the things that I did say too fast. Right, over.
Great. Thank you, Mark. Appreciate it. Have a great day, everybody. Thanks.